Okay, so let's talk about the individual parts of cells. In general, eukaryotic cells. So first of all, let's start with the cell wall. This usually happens more in plants and fungi. It's really strong, and its function is to provide mechanical support and protection against infection. It has plasmodesmata, which are channels that allow communication between plant cells. These are examples of plant cells. This is like the interior of a cell. This is interior of another cell. And this is the plant cells, the cell wall of a cell, and the cell wall of another cell. The nucleus makes up for 10 to 20% of the cell volume, so it is pretty big, pretty obvious when seen, even in a simple picture like this one. You can tell that this is a nucleus of the cell. It contains the genetic information of the cell, so the DNA. It contains a nucleolus inside of it, so here we can see the nucleus and the nucleolus inside, which is the site of intense synthesis of rRNA. RNA is ribosome RNA. It has a nuclear envelope, which is continuous with the endoplasmic reticulum. This nuclear envelope has nuclear pores, which allow the entrance of elected proteins and the exit of RNA. The fluid-filled interior contains chromosomes. Here we can see a picture of this is inside the nucleus, and this is inside the cell, so this cytoplasm. Here is cytoplasmic filament, and here is the nuclear basket. Ribosomes. They have two subunits, the large subunit and the small subunit. It's the site of protein synthesis. I'll mention this in a later video because I've learned this in high school, but I haven't actually gotten into it in university yet. I didn't need to yet, but I'll probably make a video about it later. It's composed of rRNA and proteins. It's only functional when attached to mRNA. There are free ribosomes floating around in the cytoplasm, and there are ribosomes attached to the endoplasmic reticulum, forming the rough endoplasmic reticulum. Usually what happens is we have this large subunit, and then mRNA is attached to it. Then comes the small subunit, gets attached to it, and the mRNA, the ribosome goes along the mRNA, reading it and translating it into the protein, and then forming the protein, obviously. We have the ER, the endoplasmic reticulum. Here's a picture of it. We can see that the ones that have the circles, these circles are representing the ribosomes. This is the rough endoplasmic reticulum, and the one that's smooth, that doesn't have any of those things in its surface, is the smooth endoplasmic reticulum. This is basically composed of much fuller inner membranes, which are called cisternae. The rough endoplasmic reticulum is involved in protein synthesis, whereas the smooth one is involved in lipid synthesis and chemical modification of substances. Proteins may be exported, sent to lysosomes or vacuoles, or it can also become a part of the plasma membrane. Here we can see how proteins go from the endoplasmic reticulum to the Golgi apparatus. So we have here the rough endoplasmic reticulum. We can see the ribosomes here. Protein is formed on the inside. Then a section of the membrane of the endoplasmic reticulum forms a vesicle, so it folds inwards, forming the vesicle, detaches from it, and migrates to the Golgi apparatus, where it fuses. The vesicle fuses with the Golgi apparatus membrane, and the proteins are carried inside the Golgi apparatus. This is necessary because the Golgi apparatus receives proteins from the endoplasmic reticulum and modifies them. So basically it concentrates them, packages them, and sorts them out, and then sends them to their destinations in secretory vesicles. This is a picture of the Golgi apparatus. So we have, this is the cis phase, where this is where the vesicles come in from the endoplasmic reticulum. This is its inside, so like the middle part of it. It's called the medial. And then this is the trans phase, where secretory vesicles are formed and then sent off to send proteins to their destinations. Lysosomes. This is the anatomy of a lysosome. It has a lipid bilayer, as you can see. It exists in animal cells, basically. It's involved in intracellular digestion. So basically it breaks down proteins, lipids, nucleic acids, and carbohydrates inside the cell. It can either be done by phagocytosis, which fuses with where phagosomes fuse with primary lysosomes and secrete a secondary lysosome, or by autophagy. So this is recycling of organelles, for example, mitochondria. Mitochondria. These convert stored energy into ATP. This is a picture of a mitochondria. Let me just enlarge this a little. It has a smooth outer membrane, which is permeable, and then it has the intermembrane space, which is a space between the outer membrane and the inner membrane, and the inner membrane folds in, forming cristae, which is selectively permeable. And then it has the matrix on the inside. It contains enzymes, ribosomes, and DNA. It's involved in cellular respiration, so oxidative phosphorylation in the inner membrane. It's also involved in synthesis, modification, and breakdown of other molecules, for example, some hormones. It divides by itself, asexually, and it is semi-autonomous. Now, mitochondria, as well as chloroplasts, are thought to have been, and I'll talk about this later when I'm talking about endosymbiosis. Chloroplasts. These are pictures of chloroplasts. Okay, sorry. Let's see better. It has an outer membrane and an inner membrane, so there is also a space between the outer and the inner membrane. It is made up of granum and thylakoids, and the inside of it is called the stroma. So instead of cytoplasm, it's the stroma. Its site of photosynthesis in its plant cells. It is larger and more complex than mitochondria, but they are often associated to one another in terms of their structure, things like that. And I'll explain more about it in endosymbiosis again. It has a double outer membrane. The stroma contains enzymes, ribosomes, and DNA. Again, it's semi-autonomous and reproduces asexually, which is why it's often connected to mitochondria. Plastids. These are specialized plant organelles, and they derive from unspecialized protoplastids. For example, chromoplastids. Chromo comes from color. It basically gives food and flower their coloring. So, for example, oranges, they're, orange because of this chromoplastid. Cytoplasm. As I mentioned before, cytoplasm is basically cytosol and organelles. It's the site of metabolism, where energy is used to synthesize materials, and also catabolism. It's the breaking down of materials to use energy and generate building blocks for construction of macromolecules. The cytoskeleton. This is probably going to be a complicated part. The cytoskeleton is basically made up of protein fibers that support the shape of the cell. These are its functions. It supports the shape, it anchors organelles, it acts as tracks for motor proteins, and interacts with extracellular structures, anchoring the cell. It is dynamic, which means that the fibers can spontaneously assemble and disassemble. It is made of intermediate filaments, microtubules, and actin filaments, and I'll talk about each one of them. So first of all, the actin filaments, it basically looks like this. It's long, and it's two protein strands braided together with actin subunits. It's often near the plasma membrane to support and allow change in the cell shape, so it is quite flexible. It's responsible for cell movement, so the crawling, the pinching in cell division, cellular expansion, so pseudopodia, and supports microvilli in the intestine. So this is a microvilli in the intestine, and these are actin filaments supporting it. They have a positive or a negative end, and a negative end, and they usually grow towards the positive end. The intermediate filaments are the most durable cytoskeleton element. They're quite stable, so they don't break down. They maintain the cell shape, and they bear tension. They are tough, fibrous proteins twined in an overlapping arrangement, as we can see. And then the microtubules, which are pretty much like this, they are long, hollow cylinders. I don't know if you can see here, but they are hollow. They are globular proteins, so they have a negative and a positive end, like the actin. They can rapidly polymerize and depolymerize, so they have this dynamic instability. Its functions are to provide a rigid framework within the cell, organize the cell wall, distribute chromosomes in cell division, and it allows movement of the flagella and cilia of materials within the cell, and allow movements of material within the cell. It also often forms from the centrosomes in cell division and contains centrioles. This is an example of movement of a motor protein along a cytoskeleton filament for moving, for example, vesicles in the cytoplasm. So in this case, we have an actin filament. This is the negative end, the minus end. This is the positive end. It attaches to it. The motor protein, the head, attaches to it. Then it releases. It cocks its head towards there. It attaches there, pulls it back, releases again, attaches front, and that way it moves it along. It moves the actin fiber along. Also, this is another example of movement within it. The motor proteins walk along the microtubule, carrying some kind of cargo. Again, this is the negative end and this is the positive end. So I should have mentioned all of these in this video. If I didn't, please let me know, but I'm pretty sure I did.